First of all, thank you very much for the opportunity to discuss about the hepatitis C, past, present, and the future. In April '89, we published the first data regarding the hepatitis C virus, about the structure. Now we are 26 years after the moment. What's your opinion now on April 1st, 2015?
Well, I think there's been tremendous progress in the last quarter century. For me, it's a stark contrast. In the 80s, when we were working so hard, along with everyone else, to try to discover the virus, it seemed like we were facing a brick wall, and everything we tried failed for 6-7 years. So now to have identified the virus and then to be in a situation now where we can cure patients, the majority of patients with the new drugs, is fantastic progress made by the field. So finally we have drugs that can help the patients and prevent them developing liver cancer and end-stage liver disease.
How did you start your research on hepatitis C virus?
Well, we knew that there was another disease other than hepatitis A and hepatitis B. No one really knew what was causing it, so it was termed non-A, non-B hepatitis. There was some studies performed in non-human primates suggesting it could be transmitted and therefore was an infectious agent. But nobody really knew what kind of agent it was. Throughout the 70s and early 80s, it became more and more clear that it was probably some kind of virus. But these were in the days before we had technologies like PCR, where you can amplify nucleic acids to high levels to make it much easier to discover infectious agents. We didn't have that technology, and we didn't have any experimental handle on the agent itself. So it was really, experimentally, it was like trying to find a needle in a haystack at night. We were groping around along with everyone else, but using genetic engineering technology and a lot of hard work, eventually we were able to find the virus, which we now call hepatitis C.
There was an eureka moment during your research?
Well, that's an interesting question. I get asked that a lot. The answer is no. There wasn't one moment. We were so used to finding molecular clones that we thought might be from Hep C, and then when we tested them repeatedly, they fell by the wayside. So we got used to that process. And then finally, we got a clone called 511. And we did one round of tests and it passed, and then we did another round, it passed, and then, oh, is this real? And then we did third and fourth and fifth testing, and it was almost like I was dreaming. It was almost like I had to literally pinch myself. Have we really found it? So it was a gradual process of discovery. And you know, I should add that work was done with two colleagues in the USA, Dr. Quilliam Chu and Dr. George Kuo, who were key colleagues and collaborators, without whom we would not have discovered the virus. And it took us 7 years to identify it.
And another 26 years until the treatments for the hepatitis C are available?
Yes, I thought it would be a lot quicker to develop new drugs against hepatitis C. Back in '89, I thought maybe 10 years or 15 years. As you said, it's taken 25 years. I think the reason for that is a lot of the drugs that were potent against Hep C turned out to be toxic to humans. So many clinical trials failed, not because of lack of activity against the virus, but because of toxic side events.
I want to detail a little bit your collaboration with Professor Houghton. I discussed with him and he told me that at the moment you came to his office and laboratory and asked about the non-B hepatitis research that Professor Houghton made.
Yes, yes. Yeah, Harvey Alter worked at the National Institute of Health in the USA, and he was one of the people that really first identified the problem of non-A, non-B hepatitis. He followed transfusion recipients at the NIH and realized they were getting a mild hepatitis, but it persisted it seemed, for their lifetime. So Harvey really was one of the first people to show that it was a chronic illness. And then he and other colleagues of his went on to show that actually it could cause serious liver disease if left untreated for long enough. And the work that I did in California, Harvey was in Washington, D.C. When we identified hepatitis C virus, as I mentioned, we gave many tests of the clone. And probably around test number 5 or 6 was to test a panel from Harvey in a blinded fashion. And he had sent that panel out to 40 investigators in the previous 10 years, and none of them were able to crack the code. And my colleagues and I were able to do that for the first time. So that was a significant step forward when we were able to diagnose his panel.
And
sorry, I just have to jump in. When you crossed your arms, you covered the mic.
Oh, sorry. Okay, I'll try not to do that.
And that was the moment when the non-A, non-B hepatitis has the name of hepatitis C.
Yes. Once in California, once my colleagues and I became convinced we had it, we then termed it hepatitis C. We had hepatitis A and we had hepatitis B, so it wasn't very creative to call it hepatitis C. Yes, so...
What was the reaction of the scientific community in '89 when you stated the hepatitis C virus existence? What your colleagues?
Yes. Well, I think any breakthrough, the scientific and medical community has to confirm it themselves. And so after our publications in Science in 1989, many groups from around the world sent us coded sera samples from their own patients, asked us to use our diagnostic, and we sent the results back to them, and then they said, yes, you correctly diagnosed the non-A, non-B hepatitis cases. And so quite quickly the field and labs all around the world accepted that we had identified hepatitis C virus.
Have you rated at that time of HCV virus discovery the potential impact of your discovery for the public health?
Yes, well, one of the reasons I worked on it for so long is that I realized from Harvey's work, and we had a very good collaborator also from the Centers for Disease Control, Dr. Daniel Bradley, who we collaborated with throughout our work. From these individuals, I knew that non-A, non-B hepatitis was a serious problem. Around the world, if you had a blood transfusion in the 70s and early 80s, there was a 5 to 10% chance that you would come out of the hospital with non-A, non-B hepatitis. And if left untreated for 20, 30 years, you might develop end-stage liver disease or liver cancer. So we were well aware of the importance, the medical importance, and that's why we spent so long working on it. We knew that if we were able to discover it, it would be extremely useful to patients and clinicians.
After you published the data in '89, what were your idea to continue your work? Was the development of the vaccine, of the drugs, or both?
Yeah. After the discovery of the hepatitis C virus in 1989, our first objective was to develop blood tests so that clinicians can diagnose their patients and also to develop blood tests to protect the blood supply around the world. And so our blood tests resulted in the complete protection of the blood supply. Now it's unheard of to find a single case of hepatitis C after blood transfusion. So whereas beforehand there was a 5 to 10% chance of contracting Hep C following a blood transfusion, now it's 0. That was our first objective, and then after a few years we successfully developed blood tests, and then we started to try to identify targets for drug development. So we and other groups identified the polymerase encoded by the virus, a protease, and other groups identified a protein called 5A that is very important in the viral life cycle. So then there was tremendous effort by research labs and companies throughout the world to develop drugs specifically attacking the virus. And unfortunately it took a long time because a lot of those drugs were toxic to humans. But now we're in this very good situation where we have very potent, very safe drugs to cure virtually all patients. Now I would say the remaining two challenges in the field are to get the drugs to all the patients and all the carriers before they develop severe liver disease. And secondly, to develop a vaccine, because the World Health Organization estimates there's 2 to 3 million new infections every year around the world. So we need drugs to treat the carriers, but prevention is better than cure, so we need a vaccine. So in my lab here at the University of Alberta, we are developing new drugs, novel drugs against Hep C, and we're also developing a vaccine that we want to test in the clinic in a couple of years' time.
Why is so difficult to kill the virus, hepatitis virus C?
Well, viruses evolve with their host, and many viruses cause acute infection and then the body can eradicate them. But some viruses have adapted so well to the host by genetic mutation and adaptation that they can survive for the lifetime of the patient. And HIV is one example, and hepatitis C is another example. And hepatitis C virus, I always think, is the most clever virus because HIV persists by integrating into the genome of our human DNA. Hepatitis C doesn't do that. It's an RNA virus, so it doesn't integrate, but it still persists. So it has a number of very clever mechanisms. It inhibits our immune response. Many aspects of the immune response to it. There's evidence that it might be in parts of the body that are protected from the immune response. And there are probably mechanisms that we don't even understand today. But it's a very clever virus, very smart, but finally we've outsmarted it.
That's why probably some people have the virus, but they don't develop the disease. And on another way, the treatments, they are treated.
Yes.
For hepatitis C, they clear the virus and then they relapse.
Yes. Well, that's a very interesting area. People infected with the same virus will develop different symptoms. And so the actual human genome will decide, essentially, if it's a serious disease or not. If the immune response in one individual is strong enough, the virus can be eradicated without any drugs. But unfortunately with hepatitis C, that occurs only in a minority of patients, around 1/4 of infected patients. The remaining 3/4 will develop a variety of symptoms depending on the human genetic makeup. Some people will develop rapid severe liver disease, including liver cancer, whereas others, even though they're persistently infected for life, have very mild disease. And one of our challenges is try to identify which patients will develop serious disease the fastest.
And how can we do that? How can we identify those patients?
Well, a couple of years ago, there was a breakthrough with work that showed that an interferon, IL-28B, it's a special kind of interferon, a lambda interferon, is playing a very key role in how the human responds to Hep C. If you have a certain set of mutations in IL-28B, you're more likely to resolve the virus spontaneously, or if you become a carrier, you're more likely to eradicate the virus upon treatment with drugs. So we know that genes, various human genes, play an important role in the development of disease and response to therapy, but we don't have all the answers. And I think it's very important with the new drugs that are now available to treat all of the infected carriers as quickly as possible. Economically, that's a huge challenge for healthcare systems all over the world, but if we only treat those patients with severe liver disease, we will be preventing those patients dying from the virus, but other patients that are carriers will develop serious liver disease while they're waiting for therapy. So it's very important that all HCV carriers get treated as soon as possible.
I understand that the hepatitis C, the disease, it's the result of a game between our genome, every patient's genome, and the virus, C virus.
Yes.
Considering that are many subgroups and a lot of families of hepatitis C virus, can we imagine that is not just one hepatitis C, and we can discuss about, I don't know, many hepatitis C diseases?
Yes, the hepatitis C virus is a very heterogeneous virus. It's very mutable. It changes constantly. And to put that in perspective, most people are aware that HIV is very heterogeneous and very prone to mutation. But Hep C is at least twice as heterogeneous as HIV. So there's more mutations, there's more types of HCV than there are for HIV. And that's one of the reasons, one of the causes for the persistence of the virus. The virus can mutate and continually adapt itself to protect itself from the immune response and to adapt to other cell types. However, fortunately, although there are some differences in outcome of liver disease depending on the genotype, the drugs that are in development today will be active when they're approved over the next couple of years against most of the genotypes, and will prevent all of the diseases that hepatitis C is associated with.
So we should expect in the, I don't know, 4 or 5 or 10 years, a more personalized approach of the hepatitis C based on the genotype? Well,
I think it's going to be difficult to personalize hepatitis C therapy because we don't know enough about all of the human genes required to predict response to drugs. And while we can get it right some of the time, importantly, we'll get it wrong some of the time as well. It's hard to predict patients that have mild disease now, what they will do 2 or 3 years from now if they're left untreated. We don't have enough knowledge to say that this patient can be left untreated for 5 years. He or she will be fine. We can predict that most of those patients with mild disease probably will be okay if left untreated, but there's a wide margin of error there. And certainly if I was carrying hepatitis C virus today, I would want to be treated straight away.
So the priority will be to treat all the patients now?
That is the priority. it's a very expensive burden for healthcare systems, but I think the drug companies need to produce more drugs, which they are. There needs to be negotiations between public governments and drug companies around the world to find a cheaper way to treat all patients. It's dangerous to have a plan where you only treat patients with the most severe liver disease. That is a good start, but very rapidly it needs to be extended to all patients that are carrying the virus, in my opinion.
But will be a problem with the cost, especially in countries like Romania with a low level of budget, of health budget. Do you have some, I don't know, ideas, advices?
I think for countries that have to be selective about who they give the drugs to, it does make sense to treat the patients with advanced fibrosis first. Fibrosis is a consequence of infection of the liver by the virus, and it's a serious indicator of potential liver disease, serious liver disease. So it makes sense to treat people with serious disease first, but as soon as possible, a mechanism needs to be found somehow to treat all carriers. Because a patient that's healthy now may have liver cancer in 3 years' time if they're left untreated. So prioritization of drug therapy is a reasonable strategy for the very near future, but very quickly the drugs have to be made accessible to all patients, all carriers.
In Romania, the prevalence is 3.3%, the prevalence of hepatitis C. That means 600,000 people are infected with the hepatitis C virus. 10% of them are diagnosed. Do you have a comment on this regarding the priority to treat all the patients with the interferon-free therapy now?
The first thing I would recommend is to have widespread community screening to detect most of the carriers of Hep C. Because if you can identify most of the carriers, there can be some simple procedures to minimize the progression of disease. For example, a hepatitis C carrier should not be drinking alcohol. And while that takes away some of the joy of living, if you have an infection of the liver, it will minimize the chance that you will develop serious liver cirrhosis and liver cancer. Secondly, there have been 2 good studies published by the United States NIH and the Japanese NIH, and both multi-site clinical studies have shown that something as simple as drinking 3 cups of coffee per day can actually slow down the progression of fibrosis and liver cancer. So these are very simple things that a newly diagnosed carrier can do to minimize their risk of serious liver disease. Thirdly, once a carrier is diagnosed, then that person needs to get therapy as soon as possible. If liver disease is mild, then given the high cost of drugs, they can probably wait 2 to 3 years, but somehow the governments of this world need to work with the drug companies of the world to make the drugs accessible to all of the carriers. The price has to be lower, but then the volume of drug use, therapeutic drug use, increases, and so the drug companies can make their profits and all of the carriers can be protected. So there needs to be a political and pharmacoeconomic solution to this enigma that we are facing.
What's your message for the Romanian decision maker, politician, people who can put in place strategy for hepatitis C?
My recommendation to the medical authorities and the politicians in Romania is to make the drugs available now to patients with severe liver disease. In 1 year's time, negotiate a price with multinational drug companies that is much lower than what it is now, so that those drugs can be made available to the patients with mild disease but which are at risk of developing serious liver disease. I think the message to the drug companies should be lower the price, but because the volume of uptake will be increased, their profits will still be strong, and at the same time they're serving all of the Hep C carrier population very well.
And the message for the doctors?
The message
the
for
medical doctors.
message for Romanian liver doctors is to treat your carriers of Hep C virus as quickly as possible, especially the ones with advanced fibrosis, and to really engage with the politicians and the drug companies to provide pressure to get these wonderful new drugs to the people that really need them. We can cure Hep C. It is the only chronic viral infection that can be cured. No other chronic viral infection can be cured. We have the drugs to do it. It takes pressure from the medical community, from the patients, from the government, and to work with the drug companies to get these drugs to the people that really need them. It's a tragedy, and it will be a tragedy if most carriers of Hep C cannot access these drugs over the next 2 to 3 years.
We can cure the hepatitis C and it's possible to eradicate the disease. But what are the conditions for the achievement of this goal?
I'm sorry, Marius, I missed that question.
What are the conditions for the achievement of the goal of eradication the hepatitis C?
Yeah, sorry, I still don't understand. Could you restate that?
We discussed about
Yeah.
the eradication of
Yep.
hepatitis C worldwide.
Oh, yes.
But what is necessary, what we need to do to achieve
For global
the-
eradication?
Yes.
Oh, yes, thank you. Yes. I think with the new drugs we now have available to treat Hep C carriers, and with the diagnostics and the blood tests, we have a real chance of really effectively controlling this virus amongst the world's population. One thing we need is to develop a vaccine. The WHO, the World Health Organization, estimates there are 2 to 3 million new infections every year around the globe. And everyone knows that prevention is better than cure, so we need a vaccine quite badly. And here in my lab at the University of Alberta and in many other labs around the world, there is a lot of effort going on to develop a vaccine for hepatitis C. Our own work here in Edmonton, we expect to be in the clinic in 2 to 3 years' time. And I am optimistic. Historically, there has been pessimism about the ability to develop a vaccine for Hep C, but I'm convinced it's possible. And the animal studies that we've done in the past support the notion that you can develop an effective vaccine for hepatitis C. I think it can be done, and we're working hard to do that. Of course, to control the virus globally, we need to use the drugs widely for all the patients, and in addition, we need a vaccine to prevent new infections.
Your vaccine action against all the strains of the virus is correct?
Yes, one of the problems facing the vaccine is that there's so much heterogeneity of the virus. There are so many different strains, as you said. But what we've shown in 2 recent publications from our lab here at the University of Alberta is that in humans where we gave a prototype vaccine, even though it was derived from a single strain, it could neutralize the infectivity of most of the strains seen around the world. So we've really revised the view of vaccination feasibility. In the old days, people thought that antibodies would be neutralizing only for a single strain. We know now that that is not true. You can generate broad cross-neutralizing antibodies using a vaccine derived from a single strain. And we've confirmed that with a recent paper we published a few months ago from our group here.
Coming back to the access to the new therapies, interferon-free therapies for hepatitis C, what will be the effects of non-implementing now the strategy of early access to the interferon-free treatments?
Every year that a hepatitis C carrier is not treated with the new drugs, there is a risk that he or she will develop some form of serious liver disease that could be deadly. So it's very important to have wide community screening to identify the carriers, first of all, so that they can be counseled to take measures to protect their livers, not to drink alcohol, to drink coffee on a regular basis, unless there's a contraindication for coffee drinking in them, such as high blood pressure, for example. Secondly, it is very important to get these drugs to every single carrier of Hep C as soon as possible. While most of the disease will be preventable if we treat patients with advanced liver disease first, there will be, because of the high prevalence of the virus, there will be many, many patients that develop serious liver disease if left untreated. So this is the problem with Hep C. It's not just that the drugs are expensive, it's that the disease is so prevalent, it's so widespread. So if you leave patients untreated, even though their livers are healthy today, they could come back in a year's time to their doctor with liver cancer or liver cirrhosis.
Given the high prevalence of hepatitis C in Romania, can you have a comment, a balance between the costs of the possible complication of the disease and the cost of the treatment?
Yes. Yes, I think if you do a pharmacoeconomic analysis, you can make several conclusions. And that is, if you try to save money by not treating most patients with the new drugs, initially you will succeed in saving some money, but there will be so many of those patients that develop liver cirrhosis and liver cancer, and then to treat those patients becomes extremely expensive, and a lot of the savings that you make initially will be lost because of all the treatment and all the specialist help those serious liver disease patients will need in the future. A liver transplantation is very expensive. And to treat a patient with liver cirrhosis needs an expert hepatologist to see the patient frequently and to monitor the patient frequently. It will require hospital stays probably. And overall, it's much more cost effective to treat all of the patients now with the new drugs.
In Romania, most of the patients who could benefit from the interferon-free therapy were infected during the communist period before '89 through transfusion and so on. And I would like to know your opinion regarding the state, the authorities' responsibility in this situation to provide the access for the treatment for the people infected not by their own responsibility, was the state responsibility. And now for the treatment, for the efficient treatment, it's also the state responsibility. That's correct?
Yes. Many millions of people around the world became infected with Hep C as a result of getting a blood transfusion in the 60s and 70s and 80s. Unfortunately, not enough was known about the virus then to be able to prevent that. A blood transfusion is usually given to save someone's life. If they don't get the blood transfusion, they're going to die from the road accident or from the surgery. So in the past, it's been considered on a risk basis acceptable to have a blood transfusion to save your life at the risk of getting Hep C. But that said, so many millions of people around the world have been infected following a blood transfusion that we owe it to them morally, and we owe it to them as a responsible government to make every effort to give the new drugs, to make them accessible to most of the carriers of Hep C. We have these wonderful drugs. They can cure the patient. For the first time, we can cure Hep C. And a patient shouldn't be living with the threat of developing a life-ending disease when there are drugs available. Yes, there's a huge economic burden to pay, but if you actually perform an analysis of cost effectiveness, it will turn out to be highly cost beneficial to the community to do it. And as I've also said before, the first place to start is community screening. You first have to identify the infected carriers. To do that, you have to diagnose most of the people that have had a blood transfusion in the past or have experienced high-risk behavior. That's where it has to start. And then once you've identified most of the carriers, then you can start to work with the drug companies to bring the prices down so that those drugs can be made available to most of the carriers.
Now are 3 or 4 treatments approved, new treatments without interferon for the treatment of hepatitis C. But in Romania, the patients are still treated with interferon-based therapy. Now, in 2015, what's your message for those patients who ask us about the new therapies and how can access them?
The new therapies do not include interferon. Interferon is quite a toxic drug. It can make patients feel very ill for a very long time, whereas the new drugs are well tolerated. And the new drugs can cure patients quickly, within 8 weeks or so. Interferon therapy is normally needed for 1 year. So I think the new drugs, I recommend the new drugs to all the carriers of Hep C virus. They can cure you very quickly with minimal side effects. Interferon is the old-fashioned drug. It's the one, it's history. It's the one that was used for a long time when we had nothing else. Now, the field has been smart enough to develop these new technologies and new drugs. They should be used. And if I was a carrier of Hep C, I would want to be treated with the new drugs, not with interferon. Interferon is also still expensive. It's not like interferon drugs are 1/10 the cost of the new drugs. They're not. They're comparable price to the new drugs. I think one important message to governments around the world is to understand that while the new drugs are more expensive than the old drugs which used interferon, if you actually cost the drugs in terms of the cost per cure, the new drugs are cheaper. In other words, the cost the government would pay to cure a patient today with the new drugs is less than the old cost of using interferon. Why? Because interferon only cured 50% of the patients.
Yes.
The new drugs cure virtually everybody.
And without side effects.
And the new drugs do not have the terrible side effects that interferon and ribavirin have, which are the old drugs.
In the second part of our interview, I would like to discuss about other scientific interests of you.
Okay.
About the virus D.
Ah.
You are the person who published the genome of
Yes.
virus D.
Yes. Yes. Well, hepatitis D was discovered by Dr. Mario Rossetto in 1977 in Torino. And he really was very clever to identify this unusual antigen in hepatitis B patients. most people would have dismissed that as an artifact, but he realized there was something different going on in some hepatitis B patients, and out of his work, the hepatitis D virus was discovered. My role in the history of hepatitis D was my lab in the USA was the first to sequence the genome and to identify what kind of genome. So we showed it was a very unusual RNA virus that actually was more similar to viruses that infect plants than normal viruses that infect humans. So it's a very ancient virus, actually, hepatitis D. So it's very interesting that it's persisted in the human population, and the way it's done that is that it needs hepatitis B as a helper virus. Hepatitis D on its own cannot infect humans. It can only infect in the presence of hepatitis B. So what does that mean medically? It means if you can prevent hepatitis B, you will not get hepatitis D. So the vaccine for Hep B also protects against hepatitis D. If you have hepatitis B and you're treated with drugs, you cannot be cured in most cases, but you can suppress the Hep B virus to the point where disease is minimal and where you will not be at risk for hepatitis D. But that said, in certain parts of the world, for example the Middle East, hepatitis D is a significant problem, and we do need drugs targeting the hepatitis D virus life cycle. And there are clinical trials underway, and they will be important drugs when they are approved, because there are some people that have high levels of hepatitis D along with low levels of hepatitis B, and the presence of the D virus in the liver can cause occasionally serious liver disease. So we do need new drugs against D as well.
And also we need new drugs against hepatitis B. And you are involved, and maybe
Yes.
you can say a few words about
Yes. Yes, hepatitis B, one of my colleagues here, Dr. Launterell, working with other colleagues around the world, played an important role in developing the first drugs to treat hepatitis B. Lamivudine was a drug that was developed to treat HIV patients, and then it was discovered by Dr. Terrell and Dr. Shinazi that it was also active against hepatitis B. And that led to the approval of lamivudine for the treatment of HIV patients as well as HBV. Today we have more potent versions of lamivudine to treat hepatitis B patients, but unfortunately those drugs suppress the viral replication. They do not eradicate it from the body. So now, with the cure that is possible now with Hep C patients, all of the drug companies around the world are refocusing on Hep B with the intention of trying to develop drugs that can eradicate the virus from the body. It will be a very hard challenge, but I think with our knowledge of how viruses work today, it's feasible, and I expect within the next 20 years for new drugs to be available to cure Hep B patients. We can do that with Hep C patients now, and we need to use those drugs now to cure Hep C patients all around the world.
So in 20 years, we will have a world free of Hep C and Hep B?
Yes. Yes, in 20 years, we will be able to prevent most forms of liver disease. Unfortunately, nature is very diverse, and infectious pathogens are very good at adapting to the human host. And there are other liver diseases, unfortunately. There are many different kinds of so-called autoimmune liver disease. Autoimmune hepatitis, which destroys the liver. Primary sclerosing cholangitis, which attacks the bile ducts within the liver. And primary biliary cirrhosis, also a disease of the bile ducts in the liver. These are very serious diseases. Fortunately, they're nowhere near as common as hepatitis B and hepatitis C. However, when they occur, the consequences are very serious. It's very hard to cure those patients. And for primary sclerosing cholangitis, we have no medicine at all that we can use to cure those patients. Many of those patients need liver transplantation because their liver becomes totally destroyed. So autoimmune liver disease is a new area in liver disease research. Recently, we've identified a very unusual pattern of cytokines and chemokines in the blood of those patients. And that's telling us that there really is a very specific kind of immune attack of the liver in those patients. And for example, we've shown in a recent paper that a chemokine that normally is associated with allergies, asthma, other kinds of allergies, is highly elevated in patients with autoimmune liver disease. It's called eotaxin, and it's a chemokine that normally stimulates eosinophils to migrate to the liver, and this is an inflammatory process. So that discovery gives us some hints on how, first of all, we can diagnose those patients better, and secondly, how we may be able to cure them by perhaps preventing the activity of that chemokine.
I'm sure that here is the center of the universe for hepatitis research. I'm sure about that. And I would like to ask you something related to your research. About the relationship between hepatitis C or B virus or D and different other disease like cancer, but not hepatic cancer. Maybe some other immune disease. Do you have some data?
Well, I think it's very likely that many human diseases that we don't understand today probably have an infectious basis. In other words, there's probably a virus or a bacterium or a mycobacterium that could be causing many common diseases. If you think about the diseases that plague the human population today, multiple sclerosis, encephalitis, schizophrenia, inflammatory bowel disease, lots of different forms of autoimmunity, it is very possible that the cause of those diseases is fundamentally an infectious virus or bacterium. So technology has advanced to the point now where it's much easier to identify an infectious agent today than it was 20 years ago or even 10 years ago. So I expect over the next 1 or 2 decades for many infectious agents to be found to be causing common human diseases. I think also there are many infections today that we cannot control. For example, influenza. We are under threat all the time from a pandemic strain of influenza. We have very poor drugs, very poor vaccines available to protect us against influenza. Every winter, young babies and toddlers fill up the hospitals with a disease called respiratory syncytial virus infection. There are so many infections, and of course that's not even considering bacterial infections. Everybody knows that many antibiotics today are not effective against the evolving bacterium population. So we have been complacent, I think, as humans for the last 30 years. We have assumed we have beaten infectious disease. We have not. We are under threat continuously, and we need to be very mindful of that. When we have new drugs to treat Hep C patients, we should use them. And we need to fund more research to delve into the infectious etiology of many human diseases.
I have 1 or 2 final questions for our interview. The first is, what will guide you in your work every day?
Well, for me, I was lucky that when I was just a boy at school, I discovered the biography of Louis Pasteur. And from that moment on, I knew exactly what I wanted to do in my life. And I still am just as enthusiastic now as I was 40 years ago. It's a way that I can contribute to, the best way I can play a role in society is by using my training and my knowledge of infectious disease to try to help all the millions of people that suffer from those. And I think that's my advice to young students around the world. Understand that there are millions of people dying from infectious disease every day. And you can play an important role in preventing that in the future.
How do you define this success in medical research?
My own success really has come from being committed to making a contribution to health research, medical research. I think you have to be very committed because it's very hard. It's like training for a marathon, you know, in a major competition. You have to train very hard and you have to work very hard, and it's painful because you usually have failure. It's very rare to have success, and so you have to be patient, you have to be persistent, you have to work with very good people. So I've always been working in the States. I work with very good people, Drs. Chu and Dr. Kuo, Dr. Bradley, Dr. Alter. Here in the U of A, I work with fantastic people in my lab and also with fantastic collaborators. We solve important problems by working together. In biomedicine, it's never just one person. It's always a team of highly committed, highly knowledgeable people. And I think keep working hard and be persistent and understand that it might take you 10 years or 20 years, but if you succeed, it's all worth it.
I can compare your story and your work with the story of Professor Houghton, who discovered the HPV virus.
Yes.
And developed the vaccine and received the Nobel Prize.
Yes.
And I also met Professor Houghton, and we made an interview a few years ago. And I would like to ask you about the Nobel Prize.
Well, I think the reason why we do our research is because we're interested in the science and we want to make a difference to the patients. So really everything else is small compared with those objectives. Getting prizes is nice. One of the problems I found, unfortunately, is when people acknowledge your success, they don't always acknowledge the whole team. And my work on Hep C was enabled by a large team. 2 colleagues that I worked with closely for 7 years, Dr. Chu and Dr. Kuo, and then collaborators like Dr. Bradley and Dr. Alter. So I think prizes are not that important, but I think if they're given, they ought to be given to all the team. And unfortunately, the Nobel Prize is limited to 3 people. And in my opinion, my humble opinion, I think the time has come to reevaluate how prizes are given. If it's a team of 2, give it to 2. If it's a team of 7, give it to 7. Otherwise, important team members are excluded, and that's not fair.
Your team is larger than 3, so we can ask to the Nobel Committee to change the rule, maybe starting from this year.
That will be nice.
Thank you very much for the interview.
Thank you. Thanks a lot, Marius. Very nice. I hope we got some important messages over to your politicians and to your medical community. It's a tough problem.